Hi class, this is your instructor, Skylar Huff, and we're now in Chapter 17. This is biotechnology. While it may appear futuristic, biotechnology has a long history. The use of yeast to produce leavened bread dates to ancient Egypt, and using fermentation to produce wine probably predates this by several thousand years. Modern biotechnology combines recent discoveries in molecular biology and genetics with more traditional biotechnologies such as selective breeding and hybridization. Modern biotechnology takes advantage of advances in a variety of disciplines. Progress in computer science, accelerated genome sequences, and discoveries in chemical engineering moved nanobiotechnology out of the realm of science fiction. So in this chapter, you all will explore modern biotechnology by first considering advances in molecular biology and genetics, such as the use of plasmids and other recombinant DNA techniques. Then we consider how biotechnology can be used to solve socially relevant problems in environmental science, medicine, and agriculture. So you will see those 7 sections. And as I see these 7 sections, I see that your publisher has made a bit of a mistake. This should say biotechnology, although it says the wrong thing. Nonetheless, let us begin. Biotechnology is not necessarily a discipline of the 21st century. For example, the initial domestication of dogs from wolves by our hunter-gatherer ancestors some 15,000 years ago and their subsequent selective breeding have produced the 180 existing dog breeds. Later, our agriculturalist ancestors began to domesticate crops, and the later development of hybrid corn in the early 20th century substantially increased yield. Recent discoveries in genetics and molecular biology have ushered in an era of biotechnological innovation and discovery. The ability to isolate and manipulate DNA revolutionized biotechnology, accelerated rates of discovery, and made possible novel applications of biotechnology. The construction of recombinant DNA, a single DNA molecule made from 2 different sources, began in the mid-1970s. By 1972, Paul Berg created the first recombinant DNA molecule when he inserted viral DNA into bacterial DNA. Building on Berg's work in 1973, Herbert Boyer and Stanley Cohen introduced genes from the toad Xenopus laevis into bacteria and showed that genes could be passed from generation to generation and were expressed in the bacteria. This conclusively demonstrated that genes from slowly reproducing animals could be replicated and expressed in much more rapidly growing bacteria. In the following 3 decades, the discoveries of Berg, Boyer, and Cohen would become the basis of biotechnologies, from whole genome sequencing to the production of insulin in bacteria. These early cloning experiments were some of the first to utilize new tools called restriction enzymes. Arising from basic research on how the host range of bacterial virus can be restricted, these enzymes recognize specific sequences of DNA and act as a nuclease to cleave the DNA. Originally identified by Werner Arber studying the host restriction phenomenon, the first enzyme that cleaved DNA at its recognition site was isolated by Hamilton Smith from the bacterium Haemophilus influenza and thus named HEND3. Arber, Smith, and Daniel Nathans shared the 1978 Nobel Prize in Physiology or Medicine for this work. The discovery of restriction endonucleases is important for 2 reasons. First, for their ability to cut DNA into specific fragments, and second, for their use in genome mapping. There are 3 types of restriction enzymes, but only type 2 cleave at precise locations, which enable the creation of recombinant molecules. These enzymes recognize a specific DNA sequence ranging from 4 to 12 bases and cleave DNA at a specific base within this sequence. You can look at figure 17.1 to see this. The recognition sites for most type 2 enzymes are palindromes. A linguistic palindrome, a word or phrase that reads the same way forward and in reverse, is such as Hannah, meaning Hannah with one N is the same way forward and backwards. So the palindromic DNA sequences read the same from 5' to 3' on one side as it does on the complementary strand. Given this kind of sequence, cutting the DNA at the same base on either side of the process staggered cuts that leave sticky ends or overhangs. These short unpaired sequences are the same for any DNA that has been cut by this enzyme. This allows DNA molecules from different sources to be easily joined together due to the short regions of complementarity in the overhangs, or those sticky ends. Although less common, some type 2 restriction enzymes can cut both strands in the same position, producing blunt, not sticky ends. Blunt cuts can be joined with other blunt cuts. So this is the figure I showed by using the eco-rye to cut right there to make that sticky end, and using the eco-rye to the right to cut and make that sticky end. What can now be done is the DNA from another source cut with that same restricted endonuclease can then be added to make that recombinant DNA molecule you see at the bottom. I would wish the publisher would have used different colors more so, but if you look at the bottom, you have that purple double helix that has now been joined by DNA ligase to that blue double helix, and that joined together by way of those endonucleases now have created the recombinant DNA molecule. To create a piece of recombinant DNA, 2 or more fragments of DNA must be joined together. Before this can happen, the fragments of DNA must be purified or isolated. A common technique to separate different fragments of cut DNA so they can be isolated is gel electrophoresis. This technique takes advantage of the negative charge on DNA by using an electrical field to provide the force necessary to separate DNA molecules by size. The gel, which is made up of agarose or polyacrylamide and spread thinly on supporting material, provides a three-dimensional matrix that separates molecules based on size. And you can see this in figure 17.2. The gel is submerged in a buffer solution containing ions that can carry current and is subject to an electrical field. The strong negative charges from the phosphate groups in the DNA backbone cause it to migrate toward the positive pole. The gel acts as a sieve to separate DNA molecules based on size. The larger the molecule, the more slowly it will move through the gel matrix. Over a given period, smaller molecules migrate farther than larger ones. The DNA in gels can be visualized using a fluorescent dye that binds to DNA. Gels can be made that are used solely to analyze the sizes of DNA fragments, or so that individual fragments of DNA can be cut from the gel, purified, and prepared for further use. The next step in constructing a recombinant DNA molecule is to combine pieces of DNA from different sources into a single molecule. The recombinant DNA can be stably replicated in a host organism such as the common gut bacterium Escherichia coli, or E. coli. The most common strategy to create a stable recombinant DNA molecule is to add a fragment of DNA of interest, for example, a gene sequence to be studied, into a bacterial plasmid. So you're seeing here the process of gel electrophoresis and how it works. I'll pause for a moment here. So from left to right, you have the power source, you have the negative cathode, you have the positive anode, you have the chamber. Within the chamber is the buffer solution. Within the buffer solution is the gel. The gel has wells. Inside of those wells, the DNA is added, and then that is how those fragments will move. Larger fragments move slower than the shorter fragments, and that is what creates that completed gel. Back to what I mentioned. So the restriction enzymes, they will cut at those specific nucleotide sequences, And as the DNA is cut at those specific nucleotide sequences, you're left with those fragments, and sometimes they've been referred to restriction fragment length polymorphisms. Nonetheless, reaction 1 has 2 fragments, one at 200 base pairs, another at 800 base pairs. From reaction 2, you have one at 400 base pairs, and the other fragment is at 600 base pairs. And at reaction 3, one fragment is at 900 base pairs, and the smaller fragment is at 100 base pairs. On the right-hand side, to visualize that very same, so once the gel has been completed, you have your ladder. The DNA ladder is used to see how many base pairs, or at least base pair length. And then you have those 1, 2, 3 differing DNA samples. The first you see at roughly 200, then roughly 800, lane 1, lane 2, 400 and 600, and then lane 3, 900, and then 100. That is how you read a gel. So the most common strategy to create a stable recombinant DNA molecule is to add a fragment of DNA of interest. For example, the gene sequence to be studied into a bacterial plasmid. The recombinant plasmid can then be introduced to E. coli, where it will be replicated and maintained. DNA fragments that have been cut by restriction endonucleases and purified using an agarose gel can be joined together using DNA ligase. DNA ligase catalyzes the formation of a phosphodiester bond between the 5' phosphate group and of one strand of DNA and the 3' hydroxyl group of another strand of DNA. This is the same reaction that is used by DNA ligase that joins Okazaki fragments together during lagging strand DNA synthesis in the cell. That refers to the process of replication, you all. 2 fragments of cut DNA have sticky ends, then the short complementary overhangs will hold the 2 strands of DNA together while DNA ligase forms the phosphodiester bond. Certain DNA ligases also join DNA fragments with blunt ends. In figure 17.3, a piece of foreign DNA is being introduced into a plasmid that has been cut by a specific restriction enzyme nuclease. The foreign DNA has been cut by the same restriction enzyme nuclease and thus has ends that are complementary to the cut ends in the plasmid. The sticky ends will hold the 2 molecules together, and the DNA ligase can form phosphodiester bonds, joining them to create a recombinant DNA plasmid. The whole process of producing recombinant DNA molecules is often called molecular cloning, and this shows such. So what you have here is the plasmids are being cut using restriction endonucleases at a specific site, and the foreign DNA ligases are added. And the foreign DNA and DNA ligase are added, So if foreign DNA is successfully incorporated into the plasmid, a recombinant plasmid is created in a process called transformation. So the plasmids can be introduced into bacteria. Spreading transformed cells on a medium containing the antibiotic ampicillin selects for the plasmid containing cells. So that way you want to get those cells that have been transformed. Although the central dogma of molecular biology indicates that the flow of information from DNA to RNA to protein, there are several instances in biology where DNA can be made using information in RNA. Reverse transcriptase, first identified in a class of viruses called retroviruses, can use RNA as a template to synthesize a DNA molecule. Retroviruses like the human immunodeficiency virus, that's called HIV, have RNA genomes, but then convert the RNA to DNA when they infect cells. Reverse transcriptase is an RNA-dependent DNA polymerase, which means it synthesizes DNA but uses RNA as a template, which means it's So reverse transcriptase is also a DNA-dependent DNA polymerase, which means it can also use DNA as a template to synthesize DNA. The DNA made from the information in RNA is called complementary DNA. Otherwise, it'll be called cDNA. So cDNA is called complementary DNA. Reverse transcriptase can be used to create cDNAs from mRNAs isolated from eukaryotic cells. This allows the analysis of only sequences that are actually used to direct the synthesis of proteins. A library is a collection, and before I go on to that, this figure shows the process. So you have the DNA template from a eukaryotic cell. It is transcribed. You get the mature or secondary mRNA transcript, meaning those non-coding regions have been removed, called introns, and you're only left with those coding regions known as exons. Excuse me. So isolation of mRNA by the addition of reverse transcriptase, and then the addition of mRNA degrading enzymes, and now you have DNA polymerase, and we're back to DNA that is double-stranded, cDNA with no introns. So there you have it. Now back to where I was. A library is a collection of many different recombinant DNA molecules from a single source that can be stably maintained and replicated in a suitable host organism. The source can be an entire genome or the cDNA produced by a specific cell type, or any population of DNA molecules you wish to analyze. Libraries are constructed by splicing together different DNA fragments of interest with a DNA molecule engineered to propagate the library. We call these specific DNA molecules cloning vectors. They include plasmids and artificial chromosomes. The choice of which vector to use for a library depends on how it will be used and on the size of the pieces of DNA in the library. Cloning vectors have several important features. One is a sequence that allows replication in a host organism, such as E. coli or yeast. 2, a selectable marker, such as resistance to an antibiotic. And 3, sequences that allow DNA fragments to be added, restriction endonucleases, or recombination sites. Once a library has been made, different techniques are used to identify and isolate specific recombinant molecules of interest. Libraries can be made that produce proteins to be studied for determining genome sequence and structure, or for isolating particular genes. A DNA library can be stored by being introduced into an appropriate host organism. Growing host cells will then amplify the library and produce many copies of the recombinant molecules that can be isolated or analyzed. Libraries can be introduced into bacteria or yeast by transformation, a process where cells are treated to make them take up the DNA from the environment. The host organism for the library depends on the cloning vector used for the purpose of the library. For small DNA fragments, bacterial plasmid work well, and each bacterial cell in the library will contain an individual recombinant plasmid. When the cells are grown in culture, the cloned DNAs are passively replicated by DNA replication. For libraries with larger pieces of DNA, such as genome fragments, bacterial or yeast artificial chromosomes are used, and the DNA library is stored in bacteria or yeast. Cells from a specific developmental time point or specific tissue can be used to isolate mRNA to use as a template for cDNA synthesis. The resulting cDNAs can then be used to construct a library that represents only the genes expressed at a specific time point or in a specific tissue. Genomic libraries made from different cells or tissues of an organism all contain similar sequences, but this will not be true for cDNA libraries. A brain cDNA library will contain many brain-specific sequences not represented in a fibroblast cDNA library, which will in turn contain fibroblast-specific sequences not represented in the brain cDNA library. Because a collection of cDNAs are made from a tissue reflects the mRNAs present in that tissue, comparisons of the collections of cDNAs produced from different tissues can be more informative. For example, comparisons made between cDNA libraries from different cell types might provide information about the relationships between the genes being expressed, the proteome, and the structure and function. The development of polymerase chain reaction, which is known as PCR, in 1993 accelerated the construction of recombinant DNA molecules and ushered and ushered in a new era of genetic engineering. PCR mimics the process of DNA replication to produce millions of copies of DNA sequence, a process called amplification, without the need for molecular cloning. A major benefit of producing DNA fragments using PCR is that the exact sequence of DNA to be amplified can be chosen. When the DNA molecules are cloned into vectors based on producing DNA fragments by restriction endonuclease digestion, the DNA fragments available are limited by the location of restriction endonuclease sites. To amplify a DNA sequence using PCR, two 20 to 25 nucleotide long DNA sequences called primers are chemically synthesized. One primer is complementary to one strand of the DNA at one end of the chosen sequence, and the other primer is complementary to the opposite strand at the other end of the chosen sequence. When the primers bind to their complementary DNA sequences, DNA polymerase can extend the two primers so that two new strands of DNA are produced. These two strands of DNA are complementary and contain the original primer binding sites. Repeating this process cyclically results in an exponential increase in the number of DNA molecules. A DNA sequence up to about 10,000 base pairs long can be amplified this way. PCR mimics the process of DNA replication, and an individual reaction is cycled through a series of steps where each step is analogous to the step in replication. 1. Denaturation. Heat is used to separate strands of double-stranded DNA. 2. Annealing of primers. Primers provide the 3' OH required for the elongation by DNA polymerase. And this is at a low temperature. Annealing is. And number 3, synthesis. DNA polymerase makes new DNA. New DNA molecules are synthesized exponentially with repeated cycles. A 25-cycle PCR produces 225 molecules of DNA from a single target molecule. When PCR was initially developed, the steps were done manually using a mixture containing the DNA to be amplified, the DNA template, and the primers for the fragment to be amplified. The mixture was heated to 95 degrees Celsius to denature the DNA, then cooled to a temperature for primers to anneal to the template. Then a DNA polymerase was added for synthesis. Because the 95 degrees Celsius denaturation step also denatures the polymerase enzyme, new enzyme is needed to be added with each cycle. As a typical PCR involves 25 to 30 cycles, this made PCR tedious and labor intensive. PCR was made practical with two innovations. First, the thermostable DNA polymerase called Taq polymerase. So it's thermostable. And the DNA polymerase is called Taq polymerase, was isolated from the thermophilic bacterium, Thermus aquaticus. This overcame the need to add new DNA polymerase during each cycle because Taq polymerase is stable at the denaturation temperature. The second innovation was the development of machines with heating blocks that could be accurately and rapidly cycled over large temperature ranges. A typical PCR is now set up in a single tube containing the DNA template, primers, nucleotides, and the thermostable DNA polymerase. Although Taq polymerase is still commonly used, other more accurate thermostable polymerases are also used. The tube is placed into a PCR machine that can quickly cycle between the denaturation temperature, the primer annealing temperature, and the DNA synthesis temperature. And you can see that here before you. 25 cycles of PCR can be performed in as little as an hour. This has made PCR one of the most common techniques for generating specific DNA molecules for a variety of uses. In addition to using a variety of types of DNA, plasmids, genomic DNA, as a template, PCR can also be performed on cDNA made from mRNA. Isolated mRNA is incubated with reverse transcriptase, then the resulting cDNA is used as a template for PCR. The combination of these two techniques is called reverse transcription, or RT-PCR. RT-PCR is useful for three reasons. First, it allows the creation of recombinant DNA molecules containing DNA copies of only exons of genes. And second, it allows the study of the structure and function of the gene products. Third, it can be used to determine relative levels of gene expression in cells and tissues. Cells often respond to changes in environmental conditions, both internal and external, with changes in gene expression. Analysis of gene expression involves accurate quantification of cellular mRNA levels. One of the fastest and easiest ways to measure relative changes in gene expression is using reverse transcription quantitative PCR. So that's RT-qPCR. This involves isolating mRNA using reverse transcriptase to convert this cDNA, and that converts that to cDNA. Then using PCR to amplify specific cDNAs using gene-specific primers. The amount of DNA produced can be measured in real time by the PCR machine. For this reason, quantitative PCR is also known as real-time PCR. Two common techniques used, both of which depend on the use of fluorescent dyes. The simplest way to quantify the amount of DNA made through PCR, part of real-time quantitative PCR, is to add a dye to the PCR reaction that binds non-specifically to DNA. If the DNA is present, the dye will bind to the DNA and fluoresce when illuminated by a laser. After each cycle PCR, the laser in the PCR machine illuminates the reaction, and detectors measure the amount of fluorescence. As the amount of DNA increases with each PCR cycle, more dye becomes bound to the DNA, and the fluorescence increases. The data can be output to a computer to plot a graph of the fluorescence versus the number of PCR cycles. Imagine two qPCR reactions using cDNA made from the same amount of mRNA isolated from two different cell types. During the qPCR part of the experiment, fluorescence might increase in one of the samples faster than the other. In the qPCR, where the fluorescence appeared faster, there was initially more mRNA for the specific gene because more DNA molecules were made as PCR proceeded. Computers can be used to quantify the relative differences between samples to investigate differences in gene expression under different conditions. So this is showing how it is used. A more accurate way to quantify the amount of DNA produced by a PCR reaction is to introduce a fluorescently labeled probe into the qPCR reaction. This probe consists of a 20 to 30 nucleotide long sequence of single-stranded DNA that is complementary to a portion of the DNA being analyzed. Probes are chemically synthesized and can be modified for different applications. In qPCR, a probe complementary to a short region of DNA of the template cDNA has a fluorescent molecule added to one end. When illuminated with one wavelength of light, the fluorophore emits a different wavelength of light that can be measured by the PCR machine. A so-called quencher molecule is added to the other end of the probe. This quencher will absorb the light emitted by the fluorophore, thus preventing detection. As long as both fluorophore and the quencher are attached to the same probe, fluorescence from the fluorophore is quenched. If the fluorophore and the quencher are separated, then fluorescence will be detected. If the probe is included in the PCR reaction, it can be used to detect the amount of DNA produced. After the denaturation step, when the reaction is cooled to the primer annealing the temperature, the qPCR probe also anneals to one of the template strands between the two PCR primers. As the polymerase extends the PCR primers, it will run into the annealed probe on one strand. As a Taq polymerase has an exonuclease activity, it can degrade the probe and complete copying the DNA template. When the probe is degraded, the fluorophore is released from the quencher and the fluorescence can be detected. The PCR cycles continue, and with every round there will be an increase in fluorescence that is proportional to the amount of DNA being made. The more template there is to begin with, the faster the fluorescent signal will increase. And this again shows the process of quantification with real-time PCR. It took 13 years from 1990 to 2003 to obtain a finished version of the sequence of the human genome, and an estimated cost of $2.7 billion. Today, a human genome can be sequenced in a few days for around $1,000. DNA sequencing requires that many copies of fragments of the genome be obtained. For the initial Human Genome Project, this involved molecular cloning of fragments of genomic DNA into vectors that were engineered to facilitate the manual DNA sequencing. Clones then had to be grown in bacteria and DNA isolated for sequencing, all of which is both labor-intensive and time-consuming. PCR overcame the need to use cloning vectors to obtain enough DNA to be sequenced. Many new sequencing technologies, called next-generation sequencing, NGS, use variations of PCR to produce the DNA that will be sequenced. Without PCR, sequencing genomes quickly and cheaply would not be possible. Since infectious agents, be they viral, bacterial, or fungal, all contain nucleic acids, PCR can form the basis of relatively rapid and extremely sensitive diagnostic tools. All of these tests require is a set of primers that will specifically amplify only nucleic acid from the pathogen of interest. For bacteria and DNA viruses, all you need are primers specific for some gene or genes in the pathogen's genome not found in the human genome. Depending on the nature of the clinical sample used, this may not even require isolating DNA. As the denaturation temperature will break open most cells or viruses. For RNA viruses, it is more complicated as it requires using real-time PCR, and handling RNA is more difficult. Most diagnostic kits used for RNA viruses begin with an RNA isolation step, using one of the commercial RNA isolation kits. The RNA is then ready for reverse transcription to produce cDNA, which is then used for the PCR reaction. This can be streamlined, but the basic outline is the same. One sidelight of the 2020 COVID-19 pandemic has been the development of more, faster, and better versions of these kinds of diagnostic kits. Individuals in sexually reproducing populations are phenotypically diverse, yet all members of a species usually have the same set of genes. Most diversity seen in populations is produced by complex interactions between genetic variation, epigenetic differences, and environmental variability. Advances in manipulating and analyzing DNA have produced new ways to study the genetic variation that contributes to phenotypic variation. Analyzing genetic variation is important because it contributes to understanding differences between individuals within a species and between species. Recent advances in DNA technology allow us to create or repair mutant alleles as we investigate gene function. It is sometimes desirable to be able to identify an individual based on a small amount of tissue or bodily fluids. This occurs during the investigation of crimes and in the identification of victims after catastrophic events. Although more sensitive techniques have been developed, forensics continues to use DNA fingerprinting as a primary technique. This is at least in part due to the large databases that have been assembled by law enforcement organizations. DNA fingerprinting takes advantage of short repeated sequences or that vary among individuals. I'll say again, DNA fingerprinting takes advantage of short repeated sequences that vary among individuals. Short tandem repeats, they're called STRs, typically 2 to 4 NT long, are not part of coding or regulatory regions of genes and mutate over generations so that the length of repeats vary. We say that the population is polymorphic for these molecular markers. A combination of these markers can be used as DNA fingerprints in investigations and other identification applications. And if you look at figure 17.8, it shows you that now. So this is a Y chromosome, short tandem repeat, and it distinguishes between men and women. So look at the top. And 1, because it is absent in women. Look at column 2, please. And then the short tandem repeat occurs in different lengths in men. depending on the number of repeats, columns 3 through 7, as you see there. A second short tandem repeat found on chromosome 12 appears in both men and women, and you can see that at the bottom. So again, I say, you have your control or the ladder. That's at the first column, column 1. And then the Y chromosome is there at the top. So at number 2, there is nothing there because a woman has nothing of that sort being a Y chromosome. And then 3 through 7 are from men there, showing the differences in their Y chromosome short tandem repeats. And as mentioned thereafter, at the bottom half, you see the control or ladder. That's the short tandem repeats from chromosome 12 and column 1. And then you see between those 6 individuals there, the differences in their short tandem repeats at chromosome 12. So this is another example of the flexibility and utility of the PCR process. Primers are designated to flank a region known to contain a short tandem repeat. These primers can then be used to amplify the short tandem repeat containing region from very small amounts of starting material. The analysis can also be done using the same technology used for automated sequencing. Since 1997, 13 short tandem repeats have been established as the standard for evidence for identification in court, and approved identification kits are available commercially. The 13 short tandem repeats form the basis of a federal profiling database called CODIS, the Combined DNA Index System. New DNA fingerprints can be compared to those of known individuals in CODIS. DNA fingerprinting has also been used to exonerate wrongly convicted individuals who have been imprisoned for years before DNA analysis was widely available, as well as to identify individuals after catastrophes. After September 11, 2001, attacks on the World Trade Center in New York, DNA fingerprinting was the only means for identifying some of the victims. After a devastating earthquake in the Republic of Haiti in 2010, DNA fingerprinting was used to reunite children with their families. Depending on its location in the genome, a change in one or a few nucleotides may or may not have an effect on phenotype. Any change to the genome that results in a phenotypic change we recognize as a mutation. For years, geneticists created mutations randomly using chemicals or mutagens. But now we can create mutations in vitro. Studying mutations allows us to better understand the normal function of altered DNA. The ultimate use of this approach is to be able to replace a wild-type gene with a mutant copy to test the function of that mutated gene. Remember that amplification of a region of DNA by PCR involves a template molecule, which is often a gene or cDNA copy of a gene, and a pair of primers used to initiate DNA replication of the template. If the sequence of one of the primers is made to be slightly different than the template sequence, then as DNA amplification occurs, most of the DNA sequences made in the PCR will end up with a specific mutation at a specific site. In this way, different mutations can be introduced into a gene or cDNA, and the effects of those mutations can be analyzed in a variety of ways, some of which we will explore here. As previously discussed, one of the polymerases used in PCR is called Taq polymerase. Unlike the polymerases used to replicate DNA in cells, Taq polymerase is not able to correct errors it makes during DNA synthesis. Taq polymerase will randomly introduce a mutation every 45,000 nucleotides synthesized. Changing the chemical conditions of the PCR can further increase the mutation rate, so that by performing repeated rounds of PCR, randomly mutagenized cloned genes or cDNA can be obtained. The effects of these mutations on gene product function can then be analyzed in a variety of ways. Although the ability to make and analyze mutants in vitro is extremely powerful, in many experimental systems it is not possible to introduce these constructs back into a living cell to assess their effects on phenotype. The technique of RNA interference allows researchers to reduce the amount of gene product in living cells or organisms. This is the equivalent of a mutation that eliminates the function of a gene product. RNA interference acts to degrade or block translation of a specific mRNA cell. in cells, which in turn reduces the level of the encoded protein. This technique takes advantage of the system in cells that has evolved to use small RNAs to control the level of gene expression post-transcriptionally. Using RNAi or RNA interference to reduce or eliminate the production of specific proteins requires the production of a short double-stranded RNA complementary to the mRNA that codes the protein. A variety of techniques exist to create double-stranded RNA and introduce it to cells or organisms. RNAi then depends on the cellular mechanism described in Chapter 16. For any organism for which we have a complete genome sequence, a library of short double-stranded RNA molecules can be created to target every gene in the genome. These libraries can be used to systematically reduce the expression of individual proteins or combinations of proteins so their roles in cells and organisms can be investigated. PCR-based mutagenesis allows the in vitro alteration of gene sequences that RNA interference allows the reduction of gene product. Both of these provide indirect ways to investigate gene function in living cells. The next step is to directly edit genes in living cells. For research, this allows a direct assessment of how changing a protein affects its function. And for the clinic, it offers the possibility to alter genetic variants that cause genetic diseases. While this was once the province of science fiction class, the CRISPR, clustered regularly interspaced short palindromic repeats, Cas9 system makes this not only possible, but relatively straightforward. Much of the study of host restriction in bacteria led to the discovery of restriction endonucleases. The study of how some bacteria could become immune to infection by a virus led to the discovery of CRISPR-Cas9 system. Some bacteria appear to have a kind of adaptive immunity that involves a complex system that has evolved to incorporate part of the viral genome into the bacteria's genome. This viral DNA can then be used to target future infection by a virus with the same DNA sequence. A bacterium uses the viral DNA to transcribe the RNA that is used as a guide by a nuclease, Cas9, to target DNA with the same sequence as the guide RNA. Once inside a cell, a guide RNA and the Cas9 protein associate can bind to a gene with sequences that match the guide RNA sequence. The Cas9 nuclease will cut the DNA where the guide RNA binds. Inaccurate repair of the cut DNA can result in small deletions or insertions in the genomic sequence that can result in loss of gene function. Alternatively, if linear double-stranded DNA is available, homology-directed repair can accurately repair the cut. This allows both restoring mutant alleles to wild type and creating mutations in genes of interest. The technology continues to be improved, with Cas9 proteins isolated from different bacteria that are smaller and thus easier to transfer. Cas9 proteins are also being engineered to make the system more flexible and efficient. This technique has been used to inactivate the genome of HIV, integrated into T cells grown in vitro, and has potential to treat genetic diseases. CRISPR-Cas9 can also be used to regulate gene expression. By constructing fusions between a Cas9 that lacks nuclease activity and a transcriptional activator or repressor, genes can be selectively turned on or off. It is also possible to create fusions between fluorescent proteins and nuclease-deficient Cas9 proteins. This could simplify the detection and analysis of oncogenes that are amplified in certain cancers. You're seeing right before yourselves now how the CRISPR-Cas9 genome editing system works. At top left, you have the Cas9 protein, the cleavage sites, the target, and the DNA with the CRISPR RNA used as a guide. So this is literally going to change the world class, meaning CRISPR-Cas9 is, or actually has already begun changing the world, excuse me. In this section, you'll explore how some technologies have already considered can be used to construct transgenic organisms. These organisms are modified by the addition of one or more genes from different species. Any alteration of a genome through techniques other than conventional breeding produces an organism sometimes referred to as a genetically modified organism, GMO. First, we will consider how transgenics are used for functional analysis of individual genes. In the remainder of the chapter, we will consider applications in medicines, environmental biology, and agriculture. So genetically modified organisms have been altered by techniques other than that called conventional breeding. Universal nature of genetic code makes it possible to put a human gene into E. coli, and then the removal or addition of a gene can help scientists understand the gene's function. So this is because the cells of organisms of different species interpret genetic information identically, and the genetic code is used to decipher the information in genes is the same code used in bacteria, archaea, and eukaryotes. So, for example, the amino acid phenylalanine will always be coded by the codon UUU, and this is true whether you have the genetic code in humans or examine the genetic code in E. coli. It is all the exact same. And clearly there are minor variations to the genetic code. However, for example, the genetic code of mitochondria is slightly different than the genetic code used to interpret nuclear genes. So knockout mice. So a clone gene is interrupted by replacement with a marker gene. So the marker gene codes for resistance to the antibiotic neomycin, and then there is the interrupted gene is introduced into the embryonic stem cells. And those cells are able to develop into different adult tissues because they are embryonic stem cells. So those embryonic stem cells are injected into the embryo early in development and implanted into a female with a receptive uterus. So you have the gene to be knocked out, and then you have that new gene to be entered into, the disrupted gene. And there you have it. So you then have those embryonic stem cells containing the knocked out genes. They are injected into a blastocyst stage embryo and then implanted into a female to complete development. Knockout mice. So here you have the heterozygous mouse carrying the knockout gene, and you have the homozygous mouse for the knockout gene. So the offspring will contain one chromosome with the gene of interest knocked out, and then genetic crosses can then produce mice homozygous with that knocked out gene to assess the phenotype. So this can range from lethality to no visible effect depending on which gene it was. And knocking mice, they have a normal allele replaced by an allele that has a specific genetic alteration. So this can introduce alleles that result in complete loss of function, partial loss of function, or even the gain of a function. These are those knocking mice. And then with conditional inactivation, so this allows the study of genes that are essential in development and also study of gene deletion at a specific time or tissue. So it uses the Cre-Lox recombination system. So gene of interest flanked by LOX sites, and you have recombination between the LOX sites, and that will remove or degrade the gene of interest. So you have the Cre mouse, and you have the LOX or floxed mouse, and then you have the Cre-Lox mouse with both of those added together. So tissue-specific knockout gene. So Cre is expressed from a promoter active only in the tissue of interest, and the gene will be removed only in the tissue of interest. And with developmental knockouts, Cre is expressed from a promoter that can be turned on when the mouse injects a specific compound. And then the gene will be removed only at the desired time. So this is a specific time or tissue knockout. Now to the genetic modification of plants. So unlike animals, it is generally not possible to target a specific gene sequence in plants. So integration of transient genes into plants is random. So DNA introduced into plants by electroporation, physical bombardment, chemical treatment, or bacterial transfer. And yes, this is real. So there's a tumor-inducing plasmid. So this commonly used vector for plant genetic engineering contains genes normally causing formation of a plant tumor or gall. So it came from Agrobacterium tumefacciens. So the gene of interest can be engineered into the tumor-inducing plasmid. And then part of the tumor-inducing plasmid, the gene of interest, integrates into the plant DNA and can be used to grow a mature plant in which all cells contain that trans gene. And that is how it works. So now to environmental biotechnology. So your text begins with biofuels, and those are fuels that are derived from biomass from recently fixed carbon sources, as opposed to derived from ancient biomass such as fossil fuels. And those recently fixed carbon sources are usually crop plants or algae. So it is renewable. It's more environmentally friendly than fossil fuels. So microalgae cultured in large open ponds or in self-contained culture systems called photobioreactors. And then species of algae are chosen for best production of lipids used to make biofuels. So this shows the process of how it works in this figure. So you have the nutrients, gas and water conditioning, sunlight, and you have CO2. You get algal growth. So once the algae is grown, you can harvest the algae, and then you can extract the oil, and then you can process the remaining biomass. And there's your algae oil. It can be refined there into biofuel. So bioremediation, which is that degradation or the metabolism of hydrocarbon pollutants by microorganisms. This is ever needed because sometimes things occur and bioremediation is needed. So the bioremediation of hydrocarbons, it is important for the breakdown of those hydrocarbon-derived fuels within the ecosystem. So it's potential to genetically engineer microorganisms to better break down the hydrocarbons. And then the products of those hydrocarbons metabolism are non-toxic. So this is why it's so good. It's because those products are not toxic. So they can be metabolized into biomass or even catabolized into CO2. So this is just one of those reasons that the microorganisms are so important. So microorganisms are important in wastewater treatment. So you have aerobic treatment, so that's bacteria and other organisms in aerobic tanks that degrade the carbon-containing compounds in wastewater. And you also have the anaerobic treatment, in which the anaerobic bacteria in sludge digesters catabolize polysaccharides, lipids, and proteins in wastewater. And I mention this because wastewater systems are of the utmost importance, because what you drink was once flushed somewhere if you're on water coming from some municipality. And it says it just as such. Wastewater can consist of raw sewage containing human and other animal fecal material, as well as industrial contaminants. So untreated release of this wastewater could wreak havoc on the ecosystem and even spread disease. So it must be treated. So primary treatment involves that physical and chemical techniques to remove the large and small particulate material. It's the secondary treatment that can involve digestion by the anaerobic or aerobic bacteria, or even both anaerobic and aerobic bacteria. This is why we still have clean drinking water on Earth, or at least one reason why. And then medical applications. So medically important proteins can be produced in bacteria using recombinant DNA, i.e., human insulin from E. coli. Yes, from that recombinant DNA. So originally, the human insulin came from pigs, though that did not bode well because it took a lot of time, and it really was not easy to do. Nonetheless, bacteria don't post-translationally process proteins like humans. In other words, whereas we go through transcription and translation, bacteria do as well, but there's no post-translation process. So certain combinations must be made. So genetic engineering can be used to make high concentrations of highly effective insulin. And it's happening every day here on Earth. So if you look closely in humans, this is what happens. You take out those non-coding regions, call those introns, and you get that post-translational modification. Well, in bacteria, by way of their culture, that does not have to occur. So you can culture those cells, purify the beta-gal insulin fusion proteins, purify the alpha and beta chains, and there you have your active insulin. And there is a lot more about this, and this is one of the major reasons that insulin ought not cost so much, I would say. So proteins that are isolated from the 2 strains of E. coli expressing individual insulin polypeptides can then be purified and mixed together to make functional insulin molecules. So when purified from other containing bacteria proteins, the insulin reaches very high concentrations. So unfortunately, at these high concentrations, the insulin molecules stick together, impairing their functions. So to solve that problem, specific codons in the insulin cDNA sequences were altered so the resulting proteins don't stick together. This allows the production of high concentrations of highly effective insulin. Let's get to fish. Fluorescent in situ hybridization. So fish uses hybridization to detect a specific DNA in complex mixture. So labeled DNA probe and DNA are mixed and denatured, and the probe finds complementary target DNA and forms a hybrid. So it detects large deletions, inversions, duplications, and translocations. So this is useful in a number of areas, even in, I guess you could say, determining whether or not embryo before implantation has genetic disorders, not to mention characterizing cancer and allowing appropriate drugs to be administered. So I'll say it again. In fish, the target DNA is a metaphase spread of chromosomes or cells with intact chromosomes. So fish allows the detection of gross chromosomal abnormalities, as mentioned moments ago. Moving on. So you're seeing now that it's referred to as fluorescent in situ hybridization because it fluoresces. You can see it as shown here. So you have the probe DNA, and then at number 1 you have that fluorescent labeling of probe DNA, so that's now fluorescent. And then you have the fish probe attaching to the DNA molecule, and now you can see it, the hybridization. DNA microarray, so you have collections of hundreds of thousands of different DNA sequences spotted onto a solid surface, the glass. So it can look for biomarkers associated with disease. It can look at mRNA to tell which genes are being expressed in certain conditions, and it can also diagnose genetic disorders and to genotype single nucleotide polymorphisms in an individual. So ancestral DNA services use microarrays with millions of informative single nucleotide polymorphisms instead of sequencing. These gene chips are some of the most important things used, and they do this very quickly. So infectious agents, bacteria, fungi, and viruses, they contain nucleic acids and therefore can be identified using PCR. Only primers for pathogenic specific genes are required, as earlier mentioned, and then RNA viruses are more complicated as requires real-time PCR and handling of more delicate RNA molecules. Diagnostic kits have been developed to be faster and more accurate in response to that COVID-19 pandemic. Immunoassays. So they allow the detection of a molecule through the use of an antibody. So antibodies are made commercially against viruses and bacteria, and an antibody against a virus is coated on a plate and incubated with a sample from a patient. So bound virus is detected by using a second antibody with a fluorescent or enzymatic label. So here you have that well that's coated with the antibiotic specific to the viral antigen. If you look closely at the bottom, those are your antibodies. So upon incubation, you wash, you add your detecting antibody, you wash, you add your substrate, you intubate, and then you read your plate. So that's, to me, very, and I mean very interesting. And then new vaccine technologies. So traditional vaccines were inactivated or weakened/dead attenuated viruses. New technologies include nucleic acid, a viral vector, and subunit vaccines or recombinant vaccines. So each type produces a protein or a portion of a protein to act as an antigen and stimulate the immune response. So note, it says to act as an antigen. So it's not the antigen. It acts as that antigen, and that is what stimulates that immune response. Technologies have been used in gene therapy experiments and in development as vaccines for years. So DNA and mRNA vaccines for SARS-CoV-2 are based on the spike protein of the virus. Hence, they can be identified upon infection. So gene and cell therapies. So you have the introduction of the functional to replace the non-functional one, replacing stem cell population with the correct genetic defect. using small RNAs to target expression of a specific gene, and using a patient's own modified T cells to attack those cancer cells. Now let's get to agriculture and how it is applied. One big one is called herbicide resistance. I won't spend a long time on this, but I'll spend some time. So broadleaf plants have been engineered to be resistant to the herbicide glyphosate. So the benefits from such crops are resistant to glyphosate, do not have to be tilled, i.e. the soil hasn't been turned over because to prepare for planting. So now they can just use a no-till drill. And there you go, no-till drill it in, spray with herbicide, and you're done. So that single herbicide instead of many types. And one other benefit is that glyphosate breaks down in the environment. So I don't know how many people know much about this, but in the United States, 90% of the soybeans grown is glyphosate resistant. And some just call it being, I'll use the term glyphosate, I won't use the brand name, but glyphosate resistant, i.e. that is genetically modified soy. And that's 90% of the soy grown in the United States. So the way it happens, and I'll be very brief here. So glyphosate is a powerful biodegradable herbicide, and it kills most actively growing plants. So it works by inhibiting an enzyme called 5-enolpuryl-ribosylglutamate 3-phosphate. Yeah, it's called EPSP. That's how it works. And that's the enzyme, EPSP synthase. So plants require this to make the aromatic amino acids. So again, I say glyphosate works by inhibiting the enzyme EPSP synthase, which plants require to make aromatic amino acids. So animals do not make aromatic amino acids. Rather, we obtain our aromatic amino acids from our diet. So animals living in and around crops or in waterways receiving crop runoff are unaffected by glyphosate. So to make glyphosate-resistant plants, scientists use that TI plasmid to insert extra copies of the EPSP synthase gene in plants. And if you think back with me, you learned about that tumor-inducing plasmid. So they're using the tumor-inducing plasmid to insert the extra copies of the EPSP synthase gene. So these engineered plants produce 20 times the normal level of EPSP synthase, enabling them to synthesize the aromatic amino acids and grow despite glyphosate's inhibition of the enzyme, which you're seeing right here. So these advances now allow farmers to plant these plants and not have to weed them, because of course they just spray the glyphosate and they're weeded. And they can even use the airplanes. I mean, you may have heard of a crop duster. Bingo, crop duster plants sprays the plants with glyphosate, kills all the weeds. So 6 important crop plants that have been modified to be glyphosate resistant are corn, cotton, soybeans, canola, sugar beet, and alfalfa. So the use of glyphosate resistant soy is very popular. It accounts for about 60% of the global area of transgenic crops, and that's with soy. And the other commercially important crops that are there are barley, rice, wheat, and they have all been made resistant to a range of other herbicides. Keep it in mind, BT crops. So insecticidal proteins have been transferred in the crop plants to make them pest resistant. BT toxin from Bacillus thuringiensis. So use of BT corn is the second most common genetically modified crop globally. And we'll get in a moment to stacked crops. So that means that they're both glyphosate and BT producing. For instance, many commercially important plants are sensitive to insect pests, and thus the usual defense against has been insecticides. So over 40% of the chemical insecticides used today target against the boll weevils. Hence we now have the BT crops. Golden rice. So rice that has been genetically modified to produce beta-carotene, which is a precursor to vitamin A. So it's converted in the body to vitamin A, which is deficient in millions of children worldwide. For two reasons, it's interesting. So it introduces a new biochemical pathway in the tissue of transgenic plants, and it could not have been done by conditional breeding, as no rice cultivar produces these enzymes in the endosperm. And it's available for free with no commercial entanglements. And I would say here, it's in part due to the World Health Organization. So it ensures, or at least helps, to prevent blindness in children, and that's needed. So golden rice was originally constructed in a public facility in Switzerland and made available for free with no commercial incentives. As I mentioned, since its creation, golden rice has been improved by public groups and by industry scientists, and these improved versions are also being made available without commercial strings attached. That's very important. No commercial strings attached. Now let's get on to the social issues with this and end the chapter. So adoption of genetically modified crops has been resisted in some areas for the following reasons. A, crop safety for human consumption. But then again, there is no documentation of adverse effect on human health. And I'll kind of watch what I say here. We can do more in class. But given this, in the United States, this issue has been settled for the crops that have already been mentioned. A large amount of transgenic soy and corn is consumed in this country. And yes, there remains a perceived risk of allergic reactions and longer-term effects, although there is no documentation of the adverse effects on human health. So existing crops will be monitored for adverse effects, and each new modification will require regulatory approval for human consumption. Secondly, the movement of genes into wild relatives. I'm referring to plants becoming, I guess you say, resistant to glyphosate, so to speak. So gene flow can occur between related species through hybridization, and yes, it happens already. In the United States, at least 15 weedy species have hybridized with crop plants. And for two of the major United States crops, corn and soybean, no closely related species reproduce with them, so this is at least unlikely for them. Sunflowers, however, were first domesticated in the United States, and the risk for transgene moving into a weedy population is a concern. So one study revealed that 10 to 33% of the wild sunflowers had hybridized with domesticated varieties. So in the case of herbicide-resistant transgenic crop plants, the repeated application of a single herbicide over a number of years creates selective pressure on weedy species, and over time, the weeds with resistance to the herbicide will increase in frequency, and the herbicide becomes ineffective. So almost 200 weed species worldwide have acquired resistance to one or more herbicides. So management practices that reduce herbicide use and vary the herbicides used can slow the appearance of herbicide resistance. So yes, it would also likely lead to the loss of biodiversity and more weedy species on Earth. Class, this has been your instructor, Skylar Huff. I hope that you have learned lots. And if you have any questions or comments or want to discuss anything in this chapter, let me know.